microsporangia. Today we'll discuss about the anther wall, nothing but we are not looking at the epidermis of anther. We are looking at only the anther wall, nothing but each and every sporangia containing the protective tissue as well as the nourishment tissue, as well as the wall or the layers which are responsible for the development, growth, and expelling, throwing out, splitting out of the microspores from the different sporangia. Today we'll discuss about that. And so here in the present slide, you all can able to see there are some there are the circular or structures which are showing different arrangement of the chromosome. Usually, you are going to say broadly we can say it as a metaphase stage of the chromosome in the microspores, and there you can see this what the tapetum layer, and you are going to see the nothing but the middle lamella, and finally, so which is the endothecium layer, which is not clearly visible here in this, and we'll show you in the coming slide. And coming to the first or the outermost anther wall, that is the endothecium. And usually, as I already told you in my previous class, that is in most of the textbooks, in most of the references, we are going to see the endothecium usually the unilayered. Where very rarely in most of the peculiar angiosperms or peculiar flowering plants, there you are going to see the multi-layered endothecium. But more often the unilayered endothecium you are going to see. And as we already discussed that the endothecium is going to be formed from the primary parietal cell. Let me make you very clear that any of the walls, it may be endothecium, middle layer, as well as tapetum, all the three are going to be formed by the parietal cells. It may be a primary parietal cell or the subsequent formed secondary parietal cells. But the thing is that the formation of endothecium begins very soon after the protuberance of the anther. Nothing but as there is an increase in the shape, as well as there is an enlargement, as there is an bulging of the anther from primitive only the meristematic cell containing the oblong or the quadrangular or irregularly quadrangular anther. So when as there is a, we can see the formation of archisporial cell, soon that the formation of endothecium begins. Nothing but the first primary wall, pardon, the first anther wall is going to be formed is the endothecium. That is soon after the protuberance of the anther. Nothing but as there is an bulging, as there is an increasing, as there is an enhancement of the size of the overall anther. And here the cells of the endothecium are radially arranged. Nothing but they are radially elongated. Nothing but they increase their size in this horizontal pattern, not in this vertical pattern. So nothing but what happens to cover the complete microsporangia, they are going to increase their length in a radial pattern, and the development of the what we call the endothecium can be seen till the anther dehisce, nothing but till the microspore coming out of the what we call this one coming out of the anther or a microsporangia. Till then we are going to see the existence of the endothecium. And usually what happens, these endothecium, which is are made up of polysaccharides, preferably the cellulose, and thus particular in most of the organisms, the endothecium as the microspores completes or at the late meiotic stage, they are going to lose the existence, and they are going to become a fibrous structure, and they become a dry coat. That is what we have already discussed in my yesterday's class. And this one particular endothecium, which is composed of a chemically greater amount of a cellulose, and very rarely in most of the in very few of the flowering plants, they also slightly lignified or they are slightly pectinated also. But preferably the composition of a endothecium is cellulose, that is only a polysaccharide, and very rarely in very few of the flowering plants where you can see the slight lignification as well as slight pectinification in the endothecium vertical the wall. And in most of the organisms where we are not going to see the thickening at all. So if there is any thickening of the endothecium, it is just because of a cellulose. In most of the organisms, in most of the flowering plant, there is no such thickening at all. It is maybe because of any other pattern. And the what we call the fibrous bands, nothing but when the endothecium becomes mature, when it is being said as a mature, because when nothing but it's again a consecutive steps, again simultaneous steps are occurring. As there is a development in endothecium, there we are going to see the meiotic division occurring in the microspores. When the microspore reaches active haploid pollen grains, this particular endothecium will become a dry fibrous-like structure. And what happens because of reducing in the size, what happens, fibrous structure, nothing but they are losing its shape, actual site, actual the presence. So because of that, the what we call the thickening of the wall, anther wall is going to be reduced. So this particular reduction in the wall thickening makes the what we call makes easy for the pollen grains to come out very easily after the anther dehisce. So this one, and also the next point is that when they become the very dry, like fibrous bands, they are going to become hygroscopic in nature. Hygroscopic, what do you mean of hygroscopic? Can you tell me hygroscopic? They are quite fragile to the water. They are quite fragile and they are quite easily broke down. That's what we call it as a hygroscopic. And this what a particular image I would like to show you all that. This what a original image. I mean that was nothing but the microscopic image. Not this is not illustration. This is not any hypothetical imagination of any of the part. So here you can yeah here, can you all able to see this one particular one? So this is what the extine. I mean nothing but external layer. This one, not extine, is external layer. So you are going to see the endothecium, which are you are going to see the radial pattern arranged like this, and you can see uh, some in some cases you are going to see that cells which are arranged in a two on a two tier, and the remaining we are going to see they are arranged in a single layer. And we are going to see some other components of the just see as there is an development in the microsporogenesis happening there you can see there is a the what we call uh, we can see the shrinkage of the endothecium when you compare it with first and the second. And finally you are at the this one a pollen grain, and this one again two nucleus are there, and we'll talk about pollen developments in my next class. And this one is the tetrad condition, pollen tetrad. So the pollen tetrads are to be the tetrad, particular tetrad has to be broken down so that it is going to become the haploid pollen. And that the complete explanation with regard to the pollen developments we'll discuss in my next. So this is what with regard to the first and the topmost anther wall. The next one we are going to see the middle one is the middle layer. So this usually the ephemeral nothing but they are having very short lifespan. So they are going to form next to the endothecium, and they are very they are so very soon they are going to lose their existence. And as the pollen mother cell starts undergoing a meiotic division, these middle layers start crushing themselves. Nothing but they are going to lose their presence. They are going to become, they are going to break down from the microsporangia, and usually they are multi-layered. As I already told you that in most or oftenly in most of the flowering plants, the existence of a middle layer is in multi-layered. It may be in 2, 3, 4, or 5 layers. And the most peculiar characteristic of this middle layer in some of the plants is that they also reserve some of the food material in the form of a starch. That's all. That's again some interesting characteristics in some of the flowering plants. So this is what with regard to the middle layer. And now we are at the important bit of the today's topic, that is what a tapetum. So particular tapetum, the thing is that it is going to conduct two important function. One is which is very next to the what you call the microspore developing what we call the area. Nothing but which is the very near to the spore developing area, and which is again having two function. One is protection of the internal structure, because all the walls will be rendering their performance by protecting the internal structures. The second one, important one, as the go said, it is the nourishment tissue, because it is also contains the nutrients in the form of any nutrients which are required for the development of microspore. That's what the intelligence of a plant, whatever they require, they are going to be get it from the vascular bundles. So these vascular bundles which will supply all the nutrients to the tapetum. Just see if the endothecium getting all the nutrients from the vascular bundle, then how the endothecium can make sure that the microspores are getting all the nutrients. Because my endothecium which is far away from the microspores, it may be pollen mother cell or it may be microspores. And because endothecium has to transfer the nutrients, if it is consisting of a the nourishment part, then it has to transfer from middle layer and then tapetum, and that have to reach to the microspores. So that is what the intelligence of a plant, that the tapetums which are very near to the microspores, and these are also acting as a nourishment or tissue or nourishing cells to the developing microspores. Nothing but these are functioning two aspects. One is conducting of the protection as well as providing nutrition to the growing microspores. And as well as that, they are also the source of the precursors for the pollen coat. That yesterday also I had told you in my previous class that so when as there is the pollen tetrads breaks down into haploid individual pollens, the what we call the tapetum, which is going to deposit on the outermost coat of a pollen, nothing but the substitutes for the again additional substitute for the exine also being conducted by the tapetum layer after its death, after its losing of its existence. So the mature tapetum becomes multinucleate. One more important feature is that the mitosis, that's what these are the again the somatic cells or vegetative cells, they undergo the mitotic division. So during the mitotic division, what happens usually, generally mitotic division occurs the karyokinesis after the, I mean, followed by the cytokinesis. What happens in the tapetum cells, in most of the tapetum cells, oftenly they doesn't undergo cytokinesis. They complete their karyokinesis and they become the diploid nucleated cell, and without undergoing the cytokinesis, they are going to become enlarged cell, enlarged individual single cell containing multinucleus. And that results in the formation, of course, if any cell which is having more than two nucleus are nothing but more than actual number of a diploid set of a chromosome, then it is be considered as a polyploid, right? And this is just because of the endomitosis, nothing but mitosis which is occurring inside of a cell without undergoing the cytokinesis, where the normal cytokinesis triggering molecules are absent in the tapetum, so the tapetum will not reach the cytokinesis stage, only they will complete their cycle only at my karyokinesis stage, or the formation of the restitution nucleus. Nothing but why these tapetum is becoming multinuclear. These are the four factors I'm going to explain here. One is the endomitosis. Then second one is the normal mitosis is going to fail, is not followed by the cytokinesis and the formation of a restitution nucleus. The restitution nucleus, nothing but only the nuclear division is happening. The nucleus is not at all receiving any signals, and after the formation of a diploid, two nucleus in a cell, which is that particular nucleus fails themselves very comfortable that as they are in a individual cell. And endo reduplication, nothing but endo reduplication, nothing but again it's about the chromosomal aberration, the nuclear aberration, that the in a single nucleus divides and duplicates, duplicates without causing any damage to the single cell. These are the four factors. And the next one is the synthesis of a callase enzyme. Let me tell you guys that this is what the one of the most important enzyme which is being released by the tapetum. This particular enzyme which is going to break the callose molecule. So these callose molecules which holds the four tetrads. I mean pollen in the form, when the pollen is in the tetrad form, these are the four. These are the callose is an enzyme. Pardon, callose is a chemical compound which is holding those four haploid pollen grains to become a tetrad. So here this callase is an enzyme which is going to release, which are released by the tapetum, which are going to break the callose molecule. So that what happens when this particular enzyme is going to treat on the callose molecule, the tetrads will become a freely or individual pollen, haploid pollens. And based on the behavior, after all the important characteristics of the tapetum, so based on the behavior, based on their occurrence in the different flowering plants, we have got two types. One is amoeboid type of a tapetum. Another one is a secret type of a tapetum. Is that clear, guys? Still know what I'm explaining?
Yes, sir.
Okay.
Yes, sir.
Yeah. Now, if I'm going fast at any point of time, kindly stop me there itself. Fine.
Sir, amoeboid ಅಂದ್ರೆ ಏನು sir?
That's what we'll tell you in the health in amoeboid or a periplasmodium, because
Okay,
there is no particular structure, because we can usually what happens, but they are not, they will lose very soon their actual shape. So because of more maybe because of that they have named it as amoeboid type or a periplasmodial type of a tapetum. So why this particular periplasmodial type of a tapetum, will tell you in my particular example. So these tapetal cells which fuse themselves, nothing but the neighboring tapetum cells which will fuse themselves, and they are going to become an individual tapetum. So because of that we call it as a periplasmodial each and every neighboring tapetum will contain the plasmodium, and these plasmodium will force the tapetum to get fused each other. Nothing but if, for example, let us take this, we have two tapetum, and these two are going to be joined each other and forming an individual tapetum. That is what the periplasmodium type of a tapetum. So usually such type of a periplasmodium or amoeboid type of a tapetum we are going to see in the alisma, butamus, periscantia, and typha. Hope that you people have known that as well as a periplasmodial type. So some of the points I would like to explain about how this periplasmodial type makes very interesting, and what is the difference with the another type or a secreted type of a tapetum. So here what happens during the pre-meiotic stage, this porogenous cell development. Nothing but these porogenous cell start the nothing but pre-meiotic stage, nothing but here the requirements for the formation of haploid pollen grains is started in this porogenous cells, right? So what happens during that time, the tapetal cell becomes progressively more vacuolated. Nothing but they are going to increase their size. They are at the utmost development stage, where so during that time we are going to see the formation of vesicles as well as the dictyosomes. These vesicles as well as the dictyosomes together helps in the production of a specialized enzymes, and these specialized enzymes helps in the breakdown as well as the rapid dissolution of the tapetal cell wall. You may ask why that the tapetal cell wall are going to be removed, because as the tapetal cell wall breaks down, that the components which are present in the tapetum will reach the microspore. As when it is reaching the microspore region, meanwhile they are going to complete their meiotic stage and they are going to become the tetrads, and the components which are present in the what we call tapetum, that is what the Calais enzyme which will reaches the tetrad, and they are going to break the tetrad into haploid individual pollen grains. Nothing but everything which is occurring very what we call consecutively or very simultaneously or a very programmed one. And at the end of pollen mother cell meiosis, nothing but so where we are going to, as I already told you that when the meiotic division completes, the pollen mother cell will be in a state of four-celled tetrad pollen. All the four pollen grains are attached to each other. So this has to get broke down, and that is going to be done by the Calais enzyme. This after the breakdown of a tapetal wall, the what we call the Calais enzyme will come out and it is going to react. It is going to break down the callose chemicals which are making the attachment of a four the pollen tetrads, and we are going to see finally the haploid individual pollen grain readily available, which can be thrown out for the pollination as well as the fertilization subsequent process. Where the protoplast of the fused tapetal cells move into the locules where they surround the pollen mother cell and develop in pollen grain. So what happens as they break down the protoplast, which is present in the Calais will go and break down the pollen tetrad, and the remaining components which are present in the pollen, that's what I mean the tapetum, that's what a protoplast which will come and surround the what we call this particular pollen grain. So this pollen grain will add themselves, they will become a deposition on the exine, right? So apart from that, the tapetum cytoplasm degradation as well as at the time of anthesis, we are going to see the deposition of a thin tryphin on the exine region also. So this particular exine which contains a different chemical compounds which are again a very protective one, and that particular exine contains different compounds like a sporopollenin, tryphin, as well as some of the compounds which are derived from the internal parts of the tapetum. So this is what with regard to the amoeboid or a periplasmodial type of the tapetum. And now here we'll understand the behavior as well as the characteristic feature of a amoeboid type or a periplasmodium type of a tapetum by taking an example of Arum italicum. So the person Passini and Juniper in the year 1983 have extensively worked on the actual behavior. They have reported very legitimately, very scientifically, very precisely the behavior of amoeboid type or a periplasmodial type of a tapetum, which have been present in the Arum italicum and again flowering plants. And during the pre-meiotic stage, as I already told you that during the pre-meiotic stage, we are going to see the tapetum where vesicle formation and the dictyosome formation are going to occur. So here the two-layered tapetum is going to be interconnected by the plasmodesmata. Nothing but it is going to become a periplasmodium. That's what a characteristic of amoeboid type or a periplasma type of a tapetum. As the microspore begin, it is not yet started. As they start themselves for the preparation of a meiotic stage, the tapetum are going to interconnected by the plasmodium, and they are going to become a union of a that is what I plasma in a union or individual, they're going to become a single tapetum. And when the this particular pollen mother cell at the leptotein stage, I think you might have heard about the prophase one, which is having five substages. Can anyone tell me what are those? Leptotein stage, nothing but like the first stage of the meiotic division, where what happens, the innermost tapetal cell begin to dissolve. Nothing but if the particular tapetal cell, innermost andre endothelium, middle layer, and the many layered tapetum. Innermost, nothing but which is very near to the spore microspore developing pollen mother cell or the microspores. This particular innermost tapetal is going to become dissolved, where the plasmodespota connection widen to form cytomictic channel. Cytomictic channel, nothing but so the area found between the neighboring what we call tapetum is going to be increased. For example, on the small in a nanometer size, the gap is there between the neighboring tapetum cell. That because of the dissolution in during the leptotein stage of the tapetum, we are going to see increasing the what we call the channel gap between the neighboring tapetum. That is what we call it as a cytomictic channels. And when the spores reaches the pachytene stage, there we are going to see the tangential walls common to neighboring cells are going to be disappear. Tangential wall are the walls which are near to the epidermis. And I hope that Jyoti madam at the time of explaining about the wood anatomy had explained about the tangential wall, radial wall, transverse section. Hope that ಹೇಳಿರ್ಬೋದಲ್ಲ radial longitudinal section, tangential longitudinal section, as well as some other. Yeah. So the tangential walls are the common which are neighboring to the the tangential walls neighboring to the tapetum cells are going to be disappeared. So during that time, we are going to see the attachment of a microtubules. I think you might have heard about the microtubules. These are extensively required for the during the karyokinesis as well as cytokinesis, and they are having their own role during the mitosis division. So what happens when the tangential walls are loses its existence? What happens? These microtubules are coming and lying on parallel to the radial wall. So tangential wall illa mele they are going to be seen on the radial wall, right? So they are going to be appear on the radial walls, and when they are in the microspore at the anaphase one stage, most of the radial walls now they are going to be vanish. They are going to be disappear. So overall, what happens? The tapetum initially is becoming joining each other and forming a periplasmodium type. Next, when next stage we are going to see the disappearance or breaking or losing or dissolving of the tangential wall, and next you are going to see the dissolving or disappearance of the radial walls in the tapetum. And finally, at the telophase 1, what happens? The tapetal protoplast fuses, including all the tapetums which are available, and they start penetrating towards the pollen mother cell. When the telophase 1, nothing but they are now shifting themselves to the meiotic 2 division. So when they start moving towards and they start penetrating towards the pollen mother cell, what happens? This pollen mother cell will start undergoing the meiotic division, and they are going to complete the prophase 2, metaphase 2, anaphase 2, and the telophase 2. Then what happens? Finally, it is going to release the callo. Then it is going to release the callase enzyme. This callase enzyme will break down the what you call the pollen tetrad and makes the pollen individuals. And finally, we are going to see the complete disappearance. So to as and when there is a penetration of penetration of the components of the tapetum towards the pollen mother cell, we are going to there we are not where there we are going to see the complete disappearance of any types of a wall. It may be a inner tangential wall or it may be inner radial wall are going to be completely removed. Finally, the inner layer, nothing but the components which are needed to be served for the pollen mother cell are going to be completely removed. Finally, we are going to see the tetrads when the pollen mother and the microspore at the tetrad state, we are going to see the complete disappearance of the tapetum. This is what with regard to the plasmodial or periplasmodial or the amoeboid type of a tapetum. And now we are at the second one, the glandular or a secretory tapetum. So this is what the most common type of a tapetum in the angiosperm. Okay, na? And most of the angiosperms you study, and if you go observing the tapetum, you are going to see the glandular or a secretory type of the tapetum. And here the as we already know that they are in the previous one, we are going to see the interconnection with the plasmodium, and they initially they are losing the tangential wall, then they are losing the radial wall, then they are losing the inner tangential wall. It's going to happen, and they are going to move towards the microspore. Penetration of them to the microspore can be seen. But here there is no such movement at all. They are consistent towards their actual position. But they broke down as there is a growth, as there is a development. They are going to break down, and the components which are needed for the pollen are going to be released. And the cell wall, which is quite relatively thin, because they are needed to be broke down. If they are quite tough enough, if they are quite thick enough, the breaking down again needs some more components, and because of that, the usually what happens, I feel that the glandular or a secreted type of a tapetum will be having a relatively thin wall and composed of a very low content of a cellulose. And during the premeiotic division, in the premeiotic division occurring in the spores, here in the tapetal cell wall becomes thick. Initially, the wall is going to become thick and the denser cytoplasm. Nothing but the activity of a cytoplasm is going to be increased more. And because of excessive accumulation of a cytoplasm, it has to get protected. So the wall of the tapetum is going to be increased. During that time, you are going to see the increased formation of ribosomes as well as the pro-U-based bodies. Now, you may ask, what do you mean of U-based bodies? So we'll talk about the pro-U-based bodies when I'm talking about the pollen. And I would, before that also, I would like to give some information about the pro-U-based bodies. These are the components which are associated with the ribosomes, and they are going to be found on the sporo, along with the some components of the tapetum. They are going to be what other components, they are going to be deposited on the axine, and that is what we call it as a U-based bodies. And we'll talk about the complete details about the pro-U-based bodies later on. So these pro-U-based bodies are the precursor. I mean, the precursor for the U-based bodies are being considered as a pro-U-based bodies are going to be released by the tapetum as the spores are at the stage of and at the beginning of the meiotic division. So during the meiosis, what happens? There is an further increasing the number of the pro-U-based bodies. Let me tell you that the pro-U-based bodies are the very potential components which are going to be present on the pollen grain, and they are making the pollen grains are more viable, and they are going. You might have even heard that in some of the plants, pollen grains, in the fossils, the fossil evidences of a pollen grains we have already seen. That because of the existence of a sporopollenin, because the sporopollenin is such a tough chemical compound, we are not at known which is a chemical compound needed to break that particular compound. So this sporopollenin, which is derived by the chemical agglomeration of the pro-U-based bodies, some other compounds. These sporopollen is quite tough, which cannot be broken down by heat, by chemicals, by any of the mechanism we are not going to break. Because of that tough characteristics, even till today, what we are getting the pollen grain fossil evidences is just because of the existence of a sporopollen in there in the exine structure. And so after the tetrad, what happens? These pro-U-based bodies passes through the tapetal cell membrane, and they are going to be immediately coated on a sporopollenin, and that particular component will be considered together complex as the U-based body. Nothing but the pro-U-based bodies, nothing but the compounds which are released from the tapetum cells along with the ribosomes, uh, along with some other components. They get complex with the sporopollenin, and that particular sporopollenin as well as the pro-U-based bodies will be called as U-based bodies. And finally, we are going to see the what we call the inner tangential wall thin reduction in the thickness of the inner tangential wall, and gradually it is going to be completely disappear. That gradually it is going to be broke down, and these particular U-based bodies will be settled or attached to the what we call this one pollen grain. Now we are at the now we have to discuss about the functions of a tapetum. The last slide of the today's class. What are the function? That is what we have till now studied. That is, those are the function. Nothing but the enzymes required for the making of tetrad pollen to individual pollen. That is what a callus will be released by the tapetum. Then second one is the secretion of a polysaccharide. Nothing but the what we call the nutrients which are required for the developing microspore at different stages, and secretion of a sporopollenin precursor. This sporopollenin precursor, can you tell me what is that? What is that? What is that? Sporopollenin precursor. ಈಗ ಹೇಳಿದ್ನಲ್ಲ.
Ubist bodies.
Yeah, pro-Ubist bodies. So these pro-Ubist bodies are released by the tapetum. Again, these are the precursors for the sporopollenin. Next, tapetal cells have other activities apart from these, and some other individual plants will be having some characteristic feature of their different tapetal functions. And in that, we have one example. I would like to take that Anagraceae is again a family. This the members of the Anagraceae, where the tapetal cells play a role in formation of the fine flexible threads, and that is we call it as a vicin threads. And these particular vicin threads are again the thread-like projections where we are going to see on the outer layer of the pollen. That's what on the exile. As well as the formation of a Ubist body is nothing but in the complex with the pro-Ubist bodies, as well as the sporopollenin, what that is what we call it as a Ubist bodies are completely rely on the tapetum and the formation of a pollen kit. Let me tell you that this pollen kit nothing but the chemical compounds which are required, which are present on the exile. It may be a tryphane, or it may be any of these cellulose molecule. It may be the sporopollenin precursor. It may be a pro-Ubist bodies. Altogether, these pollen kit again sometimes they are hydrophobic in nature. Nothing but they always they won't allow the water to get inside of the pollen grain, developing pollen grain, and some other components are going to be formation. The other components which are exclusively or present on the exile are going to be formed by the tapetum itself. And what happens? What this particular pollen kit is quite hydrophobic, and it contains some of the components, specialized compounds, where most of the insect or the what we call insect pollinators, they what we call they are going to be attracted by the scent or the odor. Again, that particular components are present in the particular pollen kit, as well as the formation of a pollen wall during the post-meiotic days, or post-meiotic stage also being conducted done by the tapetum itself. These are some important functions of the